speaker of this session. It is my great pleasure to introduce Laurent Gatto. Laurent has been part of the May Institute for several years now, and we are absolutely delighted to have him back. Laurent has a PhD in evolutionary biology from the Free University of Brussels in Belgium. After his PhD, he spent many years working in Cambridge University in the UK, in the Center for Proteomics, where he worked on various aspects of development of methods for quantitative and spatial proteomics, and in particular doing this within the framework of R. Currently, Laurent is Associate Professor of Bioinformatics in the Université Catholique de Louvain in Belgium, where he heads the group focusing on computational biology and bioinformatics. Very importantly, Laurent is an accomplished educator and advocate for reproducible and open access research. So Laurent is a visiting scientist in the PRIDE team at EBI. He is an affiliated member of the Bioconductor project. He is a fellow of the Software Sustainability Institute, and he is an instructor in the software and data carpentry. We are very excited to have Laurent telling us today more about his work on reproducible mass spectrometry-based research. Thank you so very much, Laurent, for joining us.
Thank you very much for the invitation. So I'll get started right away. So if any of the attendees have questions, I'll do my best to try to answer them during the talk, and maybe if there are any more technical questions, then I'll carry on answering them after the talk and the demonstration. And so if you want to follow along the slides, get access to the code that I'll be writing, or get access to the lecture notes, you can access all these here on this URL. There's a little page about today's talk with links to all the details that you will be looking for. And so I want to start by thanking Olga and all the May Institute team for inviting me, of course. It's always great to be here, even if not physically this year. But I want to thank them for the work they do, for the work in sharing some very valuable tools, but also for the work in teaching the skills to use these tools. For a reason that I think will become clear later on, I think it's absolutely essential that as researcher, we are in a position to ask any questions we want to our data. And to be able to do that, we of course need tools to ask all the questions, but also the skills to be able to come up with these questions and then answer the questions. So let me start with a motivating example as to why reproducibility is important. And that example is the inverse problem. And inverse problems are extremely hard. So in this case, we're on the left-hand side for that score percentage that students get at an exam, for example, and then on the right-hand side, the grade. The forward problem is absolutely trivial. If I get a score of 68, my grade will be B. However, the inverse problem is extremely difficult. If I get a B, what was my original score? And we have a similar inverse problem in research. From the outside, I essentially get the grade, I get a paper in PDF. But what I'm actually interested in is the score that led to that grade. And here, the score is the data, is the code, the models, the results, statistics, the software that are used to generate the paper at the end. And I'm sure some of you have already heard this or read this quote, but the paper in computational sciences, because I think it's not limited to computational sciences, is not the scholarship, right? It's merely the advertisement of that scholarship, right? The thing here on the right-hand side, this is not the actual scholarship. That's not what we are most interested in. The scholarship is everything that we have before that leads to the paper, that we should be able to access and understand as readers and researchers. And so why is reproducibility important? Well, I would say obviously for scientific reasons, right? Some people claim there is a reproducibility crisis, and we want to get access to everything that leads to the paper. But I think nowadays we also need to appreciate the importance of reproducibility for political reasons, right? In a time where the trust of the public in science, in experts, in data is not always as good as it should be, I think it's extremely important for us to provide as much clarity and reproducibility in what we do. But the point really that I want to make is that it's not only about reproducibility, or it's not about reproducibility for the sake of reproducibility. And it's not about open research just for the sake of open research, but really it's about reliable research, right? It's about trust, and openness and reproducibility are means that lead, or at least should lead, to greater trust in the research that we do and the research that we disseminate. So hopefully I have convinced you that working reproducibly is important, because that's how we can share more reliable science with our colleagues. But does it come at a cost? Well, probably yes. I mean, there is no such thing as a free lunch, right? Whatever we want to do, there is a cost. Obviously, in my opinion, that cost is worth. Generally, that cost is not financial, although sometimes it can be. It's mostly a time investment. And with the right tools and the right skills, among those that you are taught at the main institute, these costs can be minimized. But I think an extremely important point, I think, for reproducibly, beyond the actual tools that you want to learn or use to work reproducibly, it's discipline. I think discipline is really what is needed to be able to work reproducibly. And no tool will allow us to work in a reproducible way if we don't put a minimum of discipline to it. But thinking again about whether it costs to work reproducibly, maybe actually it doesn't. Maybe the possible cost is more a matter of reallocating the time. And I really like this view of whether it takes more time to work reproducibly. The first one maybe represents a kind of a typical analysis pipeline or project pipeline, where we spend some time in gathering data, analyzing data, and then we wait quite a bit of time to get feedback from our peers, and then most of the time we need to redo some analyses, and then maybe we get additional feedback, and then yet redo some more analysis. And I think the beauty of working reproducibly is that actually we're going to reallocate the analysis time at the very beginning of the project. And so it might take a little bit more time, or even if it's not a little bit of time, but even if it's taking substantially more time at the beginning, but that's time that is well invested, that we will kind of recover towards the end of the project. But so what do we mean by working reproducibly, or what do we mean by reproducibility? I think it's also interesting to think a little bit, what can we get using different tools or putting different time, different efforts into this reproducibility guidelines. And there is a kind of a classification of different levels of reproducibility. And the very first one would be aiming to be able to repeat an experiment, right? That would be to obtain the same tables, graph results, anything that is produced from an analysis using exactly the same setup, right? In the same lab on the same computer. Typically, that's if I want to repeat an analysis that I did yesterday or even a year ago. And even though that might sound trivial, it's not always the case. The second level would be to reproduce an experiment, not mine, and then again, trying to aim for the same tables, results, and so on, but this time in a different lab or in a different computer, using a very similar setup, so same type of software, reusing the same code. The third level would be the replication or replicate an experiment, and to obtain maybe not the same results, but at least similar enough results, and that's also something that would need to be defined, but then this time using a different setup. I might still be downloading the same data from public repositories. I might want to regenerate or re-simulate some of these results. And then I might, for example, re-implement the data analysis based on a description in the material method. And I'm sure that everybody that has read a scientific paper will know that that is certainly not an easy thing to do. And then finally, we have kind of the last level, which is kind of reusing information or knowledge from one experiment to reuse it, run a completely different experiment with the aim of confirming the results from scratch. And if we focus maybe a little more on the computational side, I think kind of this other tabular view is also really interesting, saying, kind of demonstrating or highlighting whether we use the same data, different data, same code, and different code. And of course, if we use the same data and the same code, it's something that is relatively easy to do. If we use different code, it's not that easy at all. And of course, different data, different code becomes most of the time extremely challenging. So, all nice and good, but in my opinion, whether we want to repeat, reproduce, replicate, or even validate, what I think really is important is that what we want to do, what we should be aiming for, is to understand what has been done and what is happening. You know, it's this understanding that is the most important, and that underlies the trust in the research we read or the research that we do. And so here, I'm not that much interested in kind of blindly rerunning something, simply to confirm that I get the same result without necessarily understanding it, which again, in some cases, is not easy at all. And I'll get back to that a little bit later. And also note that in some cases, things are not expected to reproduce, right? So if we use different versions of software, if the methods have changed, if bugs have fixed or the methods have evolved, have improved, we don't necessarily want to reproduce the exact same results that we obtained before. And so again, why is reproducibility important for myself as an individual researcher? And here I'll quote a talk that was given at the same May Institute in 2019 in Boston by a talk by Gabriel Becker. And where I think he made a really good point that reproducibility, whether computational or any, is not the point. We don't really care about reproducing something. We might actually be reproducing something that's wrong. But the goal is trust, verification, and guarantees. And so we have different level of trust depending on how much efforts we put in this level of replication that we want to attain. Is it simply trust in reporting? The results are accurately reported. If I say something, do I say the right thing? Or maybe I completely misunderstood my results. Trust in the implementation, right? For those that write code, we know that writing code is not something necessarily easy to do. So did I manage to actually write the code that does what I hope it should do? Statistical trust, right? Are the data and the methods that are used, are they appropriate, or are they still appropriate after some time? And then ultimately scientific trust, right? Are the results convincing? And do they support the claims that we are making? And so reproducibility and thus trust is really a scale from something here on the left that is absolutely not reproducible. It's essentially a black box, so we simply get a PDF of the paper and we need to take it for granted, up to scientific trust. And this is a kind of a continuous scale, and depending what we are aiming for, we might try to move towards the right here as much as possible. And so big take-home messages: reproducibility isn't binary, it's a gradient. and is very often the result of the collaboration between different people that understand different aspects of the research, different aspects of the data. And reproducibility is certainly not something that is easy to achieve. But there are many more, I think, reasons to work reproducibly as a researcher. And these are very nicely illustrated by this paper by Laurent and which are 5 selfish reasons to work reproducibly. And I think that's probably the best reason why we should be aiming to work reproducibly. It's not for kind of ideological reasons. It's simply because it's good for us. It's reproducibility that will help us in achieving certain goals. And so here are these, very briefly, these 5 selfish reasons to work reproducibly. And the first one is that reproducibility helps to avoid disasters. And what is a disaster? Well, it's typically you do an analysis and you get it wrong. And then you want to write a paper or you want to present your results. Well, that's a disaster. We all do mistakes, but if we do mistakes, at least we should be able to understand our mistakes and fix them. And without any level of reproducibility, there is absolutely no way of doing that. And reproducibility also makes it easier to write papers, because by the time I want to write, describe my results, write my conclusions, there might have been several months since I actually did the analysis. And so I don't need to kind of guesstimate or guess what I have done weeks or months ago. I can actually check it directly in my code and repeat it if I need it. Or if I don't understand figures, I can go back and understand the code to produce these figures to make absolutely sure that I know what they are supposed to demonstrate or to explain. And that not only holds for myself as an author, but it also holds for the reviewers. And if I can demonstrate to the reviewers that I provide not only the final PDF, but the data, the code, and everything that led to these results, that's a very good way to convince the reviewers that my results make sense. And reproducibility also enables continuity in our work. And that's extremely important, of course. We don't want to kind of start from scratch every time we have a new data set to analyze. We want to be able to reuse our previous work. And that's one of the things that reproducibility allows us to do. And finally, reproducibility helps to build reputation, right? Coming back to the point of trust, generally we will favor the results, the papers from authors that are known for working reproducibly, authors that provide the data, authors that provide the code. So really, working reproducibly is an excellent investment of our time. So what are the tools to work reproducibly? So I'll demonstrate some easy ways to work reproducibly using R and bioconductor packages, and I will assume that many of you are familiar or even experts with R. And my preferred way to write reproducibly is to write scripts or RMD files to keep track of what I do and be able to easily rerun the script. And the package that I will demonstrate are those from the R for mass spectrometry initiative, which I will describe a little more on the next slide. And so this is a kind of a relatively, I don't know, maybe poor man's way to work reproducibly, but it works for me. But it's possible, of course, to set up a completely pre-configured environment. And if you are familiar with Docker containers or Amazon machines instances, or I use Ranku, for example, to teach, that's also really great, because instead of simply providing a piece of code and instructions of how to install the software or how to run the code, it's possible to provide a completely pre-configured environment to fellow researchers. And in some cases, that's extremely valuable, right? If you run a 2 hours workshop, you can't spend time. What is bioconductor? I'll explain it in a second. Right? If you have a two-hour workshop, you can't spend time on installation problems, and in that case, it's really great to provide something that is already nicely packaged. But I think as researchers, investing a little bit of time to install our software is actually something really useful. And so R is a language and working environment for statistical computing. And one of the great features of R is that we can install additional packages that provide specific functionality. And Bioconductor is a project that provides packages that are dedicated to the analysis and comprehension of high throughput biological data. Bioconductor was actually initiated by one of the two original authors of R, Robert Gentleman, and it's a set of packages, packages that work coherently together to analyze high throughput biology data. And so the couple of packages that I will highlight today are some of the packages from the R4 mass spectrometry initiative. And many of these packages are already available through Bioconductor. So the links above will provide you much more information about the R4 mass spectrometry initiative. But the aim of this initiative is to provide efficient, thoroughly documented, tested, and flexible R software for the analysis and interpretation of high throughput mass spectrometry data, whether it's applied to proteomics or metabolomics. And the project formalized the long-time collaborative developments and efforts from three people, the kind of co-developers of this initiative, which are myself, Sebastian Gibb, and Johannes Reiner. And so we have been working together for many, many years now. And then I think it was in 2018, 19, we realized that it would be really great to be able to start from scratch and do some of the things we did, but do them in a kind of better way. And so there is kind of one slide of theory or background information that I need to explain before being able to dive into the code. And that's how do we see, or how do we think of mass spectrometry data when we work with the R4 mass spectrometry infrastructure. And that raw data is stored and can be manipulated using the spectral package that we'll see a little bit later. And so when you think about mass spectrometry data, we essentially think of two parts. The first one here on the left is a collection of spectra. Right, we have amyloid Z values and intensities. That's the raw data. And then each of these spectra have an annotation. And all these annotations are stored into a big table. And so each row of that table provides the mass level, the spectrum index, the acquisition number, a mass level, polarity, precursor amyloid Z, and so on and so forth for each of these spectra. Right? That's really the only thing that you need to know to be able to get started. And these different parts of a mass spectrometry data set can be stored in different modes, right? In different ways. They can be stored in memory, they can be stored on disk, using different formats. And that's something that we call backends. And then towards the end, hopefully I'll get time, I'll briefly mention backends, but that is completely transparent to the users, right? So later on, I'll be manipulating some data, and so think of these data as a collection of spectra and a big table that annotates all these spectra. And so I'll be focusing on the raw data infrastructure centered around that spectra package. So how to handle raw mass spectrometry data. If you're interested in quantitative data, that's something I will not be talking about today because I won't have time. Have a look at the Q features package. Okay, so let me remove this and bring this other window to the front. And so let me get started by simply copy and pasting this piece of code that for those that had a look is also available on the conference page. These are the lines of code that allow you to install the very latest versions of all the packages that are needed to repeat or reproduce the code that I'm going to show. Now, in a couple of weeks, once the latest version of Bioconductor is released, you won't need to install these kind of development versions, but some of the features that I want to demonstrate are not available in the kind of release, the stable version of all these packages of Bioconductor. And so the first thing I would like to do is to download some data. And to do that, I'm going to use, split my screen here. I'm going to use, oh, no, I don't want, I don't see. Oh, so I need to change a little bit because with the Zoom screen, I don't see all my screen.
Laurent, and there is a question. Can you tell everyone what R coding environment you use? Because we have talked about RStudio in the previous days, so this is something else.
Absolutely, yes. I use Emacs for all the work I do, including R coding, but this would work exactly in the same way with RStudio, right? So it's simply a window here on the right-hand side where I have some code, and I'm going to execute that code on the left-hand side. Okay, so it's really the equivalent of it's really the equivalent of RStudio. And so the first package I would like to use here is a package called RPX that will allow me to access data from the Proteome Exchange repository. And I'm going first to specify the data set that I'm interested in using the pxData function. And I'm going to say I'm interested in a specific experiment. And that specific experiment, I'm going to copy and paste the information about that paper here. And with this, we don't need it anymore. So we're going to take a subset of the data that was published earlier this year as part of a paper called RawBeans, a simple vendor-independent raw data quality control tool in Journal of Proteomics. And the identification number of that data set is PXD022816. Right? That's kind of documented in the paper, and that's what I'm going to use. And I see do you think there is an obligation to provide your raw data and code? Well, it's not for me to decide. If you want to publish your research in a journal, it would be for the journal to decide. And most of the journal will expect you to indeed publish your data, most certainly. I'm not sure about the code, but reviewers might ask it. But personally, I think, yes, there should be an obligation to publish data and code, assuming, of course, there is no private patient information. In that case, that data would need to be anonymized, right? But except for that reason, I definitely think that it's an absolute obligation to publish these. So back to my code. Let me increase the font here on the left-hand side. I'm going to create a variable, an object called px, that kind of encapsulate all the information that is available in that project. And I can use the function px files to get a listing of all the files that are available. And we can see here that we have a set of what I assume is 10 fractions. So this data has been generated on the Q Executive, if I remember well. And for each of these fractions, we have the raw data file, the binary file as was produced by the instrument, which is shown here. And that file converted to MZML, and that's what we're going to use. And then we also have the identification data as an MZID file. So let me store this into a variable called FLS that contains the name of all these files. And so what I'm going to do now is that I'm going to download these files from Proteome Exchange. And I'm going to download the MZMLs. And these are those that have the extension MZML. And so the pxcat function is a function that's going to go and check in the pxd and so on repository, or the Proteome Exchange repository for that specific project. And here I say that I want to have the first 3 MZML files. So that is fraction 1, fraction 2, and fraction 3. And I'm going to do the same thing here for the MZID. Oops. Sorry. I don't want to reinstall all the packages. So let me come here. I'm going to now download the 3 MZID files, right? Now this was extremely quick because the files didn't need to be downloaded. They were simply loaded from cache. So the RPX package tries to be clever and will not download different data sets multiple times. When the data is already available, has already been downloaded, it will simply be reloaded from cache. But so what I have here are 3 MZML files and 3 MZID files that I will be using now. But I think one way to facilitate work reproducibly is to try to store all the data together in one place or under one variable. And so when we do experiments, or when we do mass spectrometry and proteomics, we kind of think of these data as part of an experiment. And so what I'm going to do, so I'm always going to load my packages at the very top, and that the code that I'm writing now will be shared with you. I'll push it so you have it to the link through the link that is available on the talk web page. I'm going to use another package called MS Experiment. So let me load it. That I want to use to kind of annotate and combine all the information that is part of this analysis and all the information, all the data that constitutes my experiment. And so I'm going to create an object MS Experiment. I'm going to call it MSE here. MSE, and as you can see on the left-hand side, it says object of class MS Experiment. It's an empty experiment. I don't have anything yet in that experiment, but what I'm going to do is that I'm going to slowly populate it to have a coherent object, computational objects that store all the bits of that experiment that I deem important. And so the first thing that I would like now to add to this experiment are the files that I downloaded. And in this case, I downloaded these files from Proteome Exchange. But these could just as well be files that were generated in my app and that are already available on my disk. And so I'm going to say MS Experiment files to kind of store these in a coherent way as MS Experiment files. And I'm going to say that my MZML files will contain the files that are stored in my MZMLs variable here. And I'm also going to say that I also want to have MZID files. Let me check, how did I call that? It doesn't matter, you can call these the way you want. And here, oops, again. So I will go to comment this, otherwise it's going to try regularly to update my packages. So this is my way to say, well, now this experiment, or for this experiment, I have a set of files, and they are stored here as I wanted to call them MZMLs and MZIDs. And these files I now want to add. So if we look back at this MSE, if I ask, well, are there already any experiment files in this MSE? It will tell me no, there aren't any. So what I'm going to do is that I'm going to assign these MS experiment files to this experiment object. And now, before it only said that I had an empty MS experiment object, and now it tells me that as part of this experiment, I have a set of files, MZML files and MZID files. And so that's a start, right? Knowing which files belong to an experiment, again, sounds trivial, but I'm sure many of you have already had a situation where you couldn't find the files that were part of a data set. Well, that's one way to keep everything together.
Laurent, there's a couple of questions that may be good to answer now.
How would the beginner know what utilities are available? Yes. So let me see. For mass spectrometry, there is a web page for the project, and that's of course only one set of tools, right? There would be many other sets of tools, with here a list of packages that you might want to use. And for example, the MS experiment, you can click here, and this leads you to a rather detailed documentation with many more details than what we will see today that explains what you can do with these experiments. And there are some kind of non-trivial things that you can do with these experiments. And then there is also another tutorial here that provides, again, kind of use cases of what kind of things you can do with all these packages, and depending on what you want to do, which package to use. So one of the efforts we do as part of our mass spectrometer is really to provide up-to-date documentation. I don't mean to say that suddenly with all this documentation it becomes straightforward for a beginner to start analyzing their data with these packages. There is a learning curve, of course, but I guess that's the price to pay. But the documentation to make that as easy as possible is available. So what do you think the TAC replicate using mass spec? And by TAC replicate, do you mean technical replicates? And regarding all the links that I have shown are already available. So if you go on the Google Doc or the link on the conference page, you will get access. You have access to all the links and all the slides and all the code that I'm showing. And so regarding the previous question, I'm not sure what you meant, but if you meant technical replicates, I certainly believe that biological replicates are much more interesting than technical replicates, but that's more of a experimental design question, I think. And so in PX get here, to answer the other question, PX is the object that represents this protein exchange experiment. And here, PX FLS contains the list. We can look this here. FLS contains the list of all the files that are interested in. And here, I want to get not all the files, but I'm only going to look at the first 3 files. Yeah, and so technical replicates. I don't think technical replicates are the best investment of our time and money. I would certainly rather suggest to do biological replicates, as long, of course, as the technical aspects are handled, right? This, of course, assumes that the kind of running the experiment is something that is in place. And so coming back to my MSE object, well, currently I have an experiment that contains file. In a minute, we're going to add raw data, but I might want to add additional information, any information I'm interested in. So for example, if I look at the metadata, well, metadata is empty, it's an empty list, but I could say, pxt, and of course, something that would be interesting would be to store here the proton exchange identifier of that experiment, and then maybe the PubMed ID. Let me store this too here. Here, let's say PubMed ID. And so I could kind of add these things to my MSE object. And now if I say metadata MSE, it tells me that I have a PXD element and a PubMed ID element, right? So I could really add any metadata, any information I'm interested in here to very simple elements, but these could be much more complex, and some of these are described in the vignette. So very good question. So these packages only focus on data that was exported into one of the open formats, such as MZML, MZXML, and so on. Right? They do not read raw or binary files, but as long as you can convert to MZML, it will work. Yes, and so if you get this error, I assume that pxfls, that variable, was not created in your case. I'm not expecting people to type along with me. You will have all the code if you want to kind of replicate or repeat what I did afterwards. So I think it would be probably a bit of a challenge to try to repeat everything I did. Okay, let's go to the next step of our data and load the raw data. And to load the raw data, I will use another package called Spectra. And I'm going to load that package here at the top. So here in my environment chunk, I will, every time I need a new package, I will load that package there. So it's easy for me later on when I want to understand what my script did. It's easy for me to kind of go back and check what were all the packages that I needed. Okay, and so once I load the Spectra package, I have access to the new function called Spectra that will create such an object that contains all the raw data. And which files do I need to generate such an object? Well, the MZML files. And where do I have these MZML files? Well, in my experiment, I can extract the experiment files of MSE. Okay, and that's a list with 2 elements. And among that list, those that I'm interested in are those that are called MZMLs. So these are the 3 files that contain my raw data. And so given that I'm working in the frame of this experiment, I'm not going to retype file names or get them somewhere else. I would immediately use the file names that already exist in my experiment. So all the data can simply be downloaded from Proton Exchange. You're absolutely right. You can go on an FTP site and then download the files manually one by one. You're absolutely right. The advantage of doing this here is twofold. Well, the first one is that I see exactly which files I download. Okay, so there is a script, a piece of code that demonstrates which files I downloaded, which is something that you could do with discipline and write it down in your lab book, right? That would be just as good. The second advantage is that this will automatically cache your data. So if I download it once, it will not download it again if I need them, in another directory. But a lot of the things that I'm presenting here could be done in different ways, for instance, manually, and that's perfectly fine, as long as you have the discipline to work reproducibly, without using some of the tools that I present that help you work more reproducibly. So I'm going to create a new Spectra object with the files that I have already stored as part of my experiment. And I'm going to call this object SP. So here we go, here on the left-hand side. SP now contains a raw data object that contains 87,647 spectra, right? All the spectra that were present in these 3 files here that we downloaded from Proton Exchange. And now it's kind of useful to remember that slide that I showed where when I said, well, a spectrum, or sorry, no, an MS experiment is composed of two things. One big table that contains all the annotation. And what is this annotation? Well, that's something we can get with the function spectra variables. If I say spectra variables SP, it lists a vector of 34 elements. And these elements are the annotation that we have for each of these 87,000 spectra. So we have the MS level, RT for retention time, acquisition number, polarity, whether they're centroid or not, and so on. So that's all the information that is contained in that table. It's a table with 87,000 rows and 34 columns. And so spectra variables allows me to get the name of these variables, these annotations. Spectral data allows to actually get that big table. Now, it's too big to be shown, so we only see the first row, but spectral data allows me to get that big table with the annotation of all my spectra. So what kind of results am I trying to reproduce? I'm not trying to reproduce anything specific from that data. I'm going to show how to load data, how to do some quality control, some visualization of the data. I'm not trying to replicate anything specific. But the work that I'm doing now, you should be able to repeat it by installing the package. So I was saying, remember big table, spectral variables, and spectral data. That's how we can access the information in that big table with the annotations. And then there was a second piece of information in a raw data experiment, and that was the M over Z and intensities. And that's something that we can access with peaks data. So if I say peaks data, before I get to that, so SP contains my full experiment of length, 87,000, and I can very easily take subsets of this experiment. So for example, if I simply say 1 or 2, sorry, 1 to 2 here, SP 1 to 2 behaves like a list, and we'll create a new experiment with now only 2 spectra. And so if I want to access the so-called raw data, I can do that with the peaks data function. And the peaks data will actually go and collect the MZ, M over Z values and intensities. So let's have a look at the kind of M over Z and intensities of first spectrum. Here there are, right? So that's the first spectrum in that data set. So we can go and access the pieces of information that we are interested in. Now we can also visualize these. So instead of looking at the matrix here, maybe it's easier for me to say, well, plot spectra. I don't want to plot the full experiment, but only the first one. And it appeared here at the back. Here we go. So that's the visualization of the first spectrum. And here on the left-hand side, you have the actual data. Right. That was, where is it? That is a spectrum MS level 1, retention time 0.2. So that's the very first spectrum that we have in this data set. If I type any number here, and before I tried with 23822, and if I rerun this, this is an MS2 spectrum. You can see here MS2 at this retention time. So it's relatively easy to extract, and if we want to visualize these data. Well, let me now load another package very briefly. That is called SpectraVis. And with SpectraVis, what we can do is the following. We can use a function called plotly spectra. And that one will open, and it opens here, kind of an interactive way to visualize these data. So that's the same spectrum, still spectrum number 1 shown here. but it provides some flexibility. If that's what you need to do, you can kind of zoom in into specific regions of your spectrum. Right, so once you have the data, you can easily visualize it either using static or interactive visualizations. But so we have all that data, so let's try to explore it a little bit. And let's try to check, yes, if you use RStudio, it will still be interactive. This doesn't use any custom infrastructure. It uses the plotly package that also works with RStudio. And for any errors, I will look at these later on and try to debug and help you and with the installations of some of the packages. So how do you get the M over Z and the charge? Well, that's an example that we can have. The charge of the precursor, well, we could say precursor charge, sorry. Charge here is P. And this typo precursor charge, this would kind of get us the precursor, the charge of all the precursors. Of course, if we have an MS1 scan, there is no precursor, so we have an A. And so, for example, here we can see that among all these spectra, in this case, the MS2 spectra, we have close to 46,000 spectra that have a precursor charge, or MS2 spectra that have a precursor charge of 2, and then 20 or so, 30,000 of 3, and so on. So that would be precursor charge. And then if you wanted to have the M over Z values, well, you can access these with the peaks data function. So, same thing for the intensities. But so let's have a little look. So SP is this raw data object, and there is the data origin variable that tells us where do these spectra come from. And so if we look at the table of this, we see that we have more or less the same number of spectra in each file. Right? 29,000 in the first file, 28,400 in the second, and 29,300 in the third. Okay, and then something else that I find actually quite interesting is to say, well, here we have the MS levels of all these. Let me show this. Okay. Of all these data. Actually, we can maybe table of this. So we have 19,000 MS1 level scans and 68,000 MS2 level scans. And we can also check whether these are centroided. Put the centroided here. If I only look at the 6 first ones, they are all centroided. And something that I actually find really interesting is to have a table of MS level by centroided. And in this case, I realized that all my MS1 spectra are centroided and all my MS2 spectra are centroided. But very often you might have MS1 spectra that are in profile mode and MS2 spectra that are centroided. And if you have MS3, you could also kind of tell which ones are centroided and which ones are not, which is something that might be interesting for downstream analysis. But we have all this raw data, so let's try to kind of make use of these and do some visualization. And so I'm going to go back at my very first part here and load 3 other packages, ggplot2 for visualization, tidyr for some data manipulation, and then magrittr to use the pi operator. So I'm going to load these 3 packages here. And let's do some visualization. And the first visualization that I would like to do is to visualize the chromatograms. So let me add this here, the chromatograms. So chromatograms only need MS1 data. So I'm going to use the filter MS level function on my SP object to only retain MS level 1 spectrum. And so we know here that we have 19,607 MS1 spectra. And then I'm going to extract this metadata, also it's this big annotation table with the spectral data function that we have seen before. I'm going to convert it to a table, and then I'm going to use ggplot to visualize the total ion current along the y-axis along the retention time. And I want to separate this based on the data origin, which is the file where these are produced by. And so here is that figure. Let me make it wide. Right, so we have extracted the information that we see here on this figure, the retention times along the x-axis and the total ion current along the y-axis. And here we can visualize the total ion chromatograms of these three-row experiments. And we can confirm that they're actually quite similar, some difference at the beginning, but these look like very good chromatograms. Something is maybe a little bit surprising here for the second file, in green here, that's the, oh no, sorry, the third file. Somehow, apparently, there is a short time during, what is this, 1,800, 1,900 seconds, where suddenly we don't apparently have any MS1 scans that were acquired. No idea whether this is something we should be worried about or not, but at least it allows us to easily and quickly produce these chromatograms, which are a really good way to kind of QC your data. Okay. And then something else that I find really interesting when it comes to quality control. And I'm going to run this because it will take a little bit of time, and you can get the details of the code here. I'll leave this command in the code because it's not in the package yet. So we are going to only extract here the first file. Right. Filter data origin will tell that, among all the, 87,000 spectra, only keep those that belong to the first file. And then I'm going to call the compute MZ deltas function. And so what this function is going to do, it's going to first only extract the MS2 spectra. What is the environment that I use? So I use, let me type it at the very beginning. I use the Emacs software, and the kind of package that allows me to manipulate this R code and run it is called ESS. Okay, but just for your information, RStudio is really great, and so if you are not already familiar with Emacs, I would not advise you to get into Emacs just for the sake of using or using R. RStudio is really a great piece of software, so by all means use it. And so, this MZ delta, or compute MZ delta, is going to extract all the MS2 spectra. And an MS2 spectrum is supposed to represent some information about the identification of these peptides. This is a proteomics experiment. And the peaks that we see in the MS2 spectrum are supposed to reflect peptides, or the difference between all these peaks is supposed to reflect the masses of different peptides. And so this function is going to take all the MS2 spectra, only consider the highest MS2, the highest peaks in these MS2 spectra, all the fragment peaks, and then it's going to calculate the distance between all these peaks. And if what I have analyzed here, or if the data that I have downloaded actually analyze peptides, and we know that's the case, well, then I expect that all these distances, these differences between fragment peaks in my MS2 spectra correspond to the masses of amino acids. And so this function is going to calculate all these distances between all the fragment peaks of all the MS2 spectra. Well, actually, maybe it's not all, it's only a kind of certain percentage, or only the fragment peaks that are in the top quantile, or something like that. And so it's going to do that. And it looks like it's taking a lot of time, and I think that's because Zoom is taking quite a few resources. Let's see. Okay. Yeah. Okay, let's give it a little bit more time. Here we go. And so if I look at D1 plus D1 here, it's a list. And you see this is a list with all the deltas between the fragment peaks of my MS2 spectrum. And now if I visualize this, where is my figure? Where has it appeared? It has appeared here. I obtain this histogram here. Not sure, can you see my histogram?
Yes, yes, we can.
Yep. Good. Well, along this histogram, I am highlighting with kind of vertical lines the masses of my 20 amino acids. And indeed, I observe that I have an increase of these MZ deltas in this data. So that is typically the kind of histogram of mass delta distributions that I want to see. I want to see an increment in mass deltas corresponding to peptides. As many or as everybody have already had experiments that completely failed, and then you would get essentially a flat histogram, which would be a really good indication that even if you run a search engine, but maybe your parameters were not optimal, this will give you a kind of an unsupervised quality control of your MS state. And so will this work for modified peptides? Yes, it will work. You might want to also highlight the masses of your modified peptides, but this should also work for modified peptides. Yes, it's completely agnostic of the technology that you used. Okay. And so that's another type of quality control that I find really useful and that I tend to do, especially when I don't quite get the number of identifications that I'm supposed to get. It's always good to try to have a, let's say, search engine independent quality control. But so I have here my SP object, of course, and it still kind of lives outside my experiment. And so if I want to add it to my experiment, I can say spectra MSE and say SP. So now I'm going to kind of add that spectrum object to my experiment. And now if I simply print the name of my experiment, it tells me, well, I still have the files that I had before, but now this experiment also contains that many MS1 spectra and that many MS2 spectra. And I'm not going to do it now because it's not very interesting in terms of demonstration, but I could save this object, close R, reopen R, and simply load that object, and then I would get the raw data, all the files, and any metadata that I have added. So let's have a quick look now at the identification data that we had. And for that, I'm going to use a package called PSM. And then I'm going to load that package, but I prefer to put all my package loading at the top. And now I'm going to load these identification files. Where can I get these identification files? Well, with experiment files. Okay, here they are. And then $MZIDs. Here are the 3 identification files that I downloaded. So I'm going to load these into, with a function called PSM, into a variable that I call ID. And it's kind of shown here. This is an object PSM that contains information from MZID files, and it contains 74,750 PSMs. And I have 31 variables or columns that I can look at here, names, ID. So I have here these 31 annotations for all my PSMs. And so I can extract these, and I can kind of manipulate these. And one visualization that I'm going to do is the following one. So this ID variable, it says it's an object, it's a PSM, but really it's only a kind of a glorified table. And I can very easily convert it into a tibble. If you are familiar with tibbles or data frames, and here it is. So simply by saying as tibble, I convert it into a tibble, and then I'm going to feed that tibble into ggplot2 to produce a visualization. Before I say I have a question here. Yes, that is exactly what I was looking for. These MZ deltas should roughly correspond to the masses of amino acids. Yes. Without considering any information about the search engine, I kind of look at the raw data in an unsupervised way, and I just want to make sure that these deltas correspond to amino acids. And so with this piece of code, I'm going to plot the metamorphosis score, actually density of these scores, and then I'm going to color code these densities based on whether that PSM comes from the decoy database or the forward database. And this produces this figure here. And so I split these figures based on the spectrum file, the raw data file. And so that's, I think, also the kind of visualizations that are always extremely useful, not just look at the number of identifications, but here I can clearly see that those that are decoy peptides, so in blue, tend to have much lower scores than those that are in red here, which are non-decoy peptides, right? So I want to see a distribution shifted towards higher scores for my non-decoy peptides. And this is something that I did for the metamorphosis scores, but I can do exactly the same thing for the, what did I use here? The PSM level Q values, right? And so we see that we have much smaller Q values here for the non-decoy peptides. But note here that actually these identifications that I downloaded through these MZID files have already been filtered. As you can see here, we have Q values that are smaller than 0.01. So this is already our 1% data set that contains the decoy and forward peptides, but I see a clear enrichment here for non-decoy peptides. So good quality identification data. Now, of course, we could kind of look at these identification data in great detail, but in this case, in the interest of time, I could simply filter these PSMs, remove all the decoy hits, remove any PSMs that have a rank greater than 1, only get rid of the non-proteotypic peptides, and this would leave me with 3,700 high-quality PSMs. How do you make it work for TMT labeled peptides? Yeah, no, I don't think that's a problem with TMT peptides because you don't have all the single amino acids that are labeled. So the MZ delta histograms also works for kind of labeled peptides. So here I have all my PSMs, but I can use the filtered PSM to, and let me overwrite this object here, to now get a reduced set of 36 or 37,000 high confidence PSMs. Now I have my PSMs, I have my raw data, but what I really want to do is to combine these, to join the raw data and join the identification data. And for this I need, yes, so the filtering, so this is kind of one function that does lots of filtering together, but in the interest of time, I quickly use it like this. But yes, you can parameterize the type of filtering that you want to do. Is there a Google group? There is no Google group, but we have we answer to questions on GitHub, or we also use the. You're also free to ask questions on the bioconductor support forum. And this is described. Where to ask questions is described on the respective web pages. And so, what I really want to do before, because I see the time is running, is to combine these raw data and these IDs, these identification data. And so to be able to combine these, we need to have a shared variable from the raw data. And you remember, if I look at the spectra variables, these are those that I have, right, in my raw data. And then if I look at, what was it, names, ID, these are the variables that I have for my identification. And I somehow need to be able to match these. And that's not difficult to do, given that I have file names and was it spectrum file and spectrum file, right? So data storage here and spectrum file here represent the same information. The raw data file, here spectrum file, is the raw data file from which the identification comes from. And the second information is the spectrum ID. So where is it? I can't quite spot it. So I have spectrum ID here for my identification data. And I also have a spectrum ID for my raw data. Where is it? Spectrum ID. Oh, here it is. The difference, and that's the nature of the data. I have a lowercase d here and a capital D. No, I lost again. A capital D on the other side. But I have all the information that I need to be able to precisely match spectra in specific files between these two data types. And so I'm going to do that, and you will have the code, so you can look into details by creating what I call a primary key. And the primary key here is composed of the file name and then the scan number, right? So I know that the 14th element in my raw data file is scan number 14 out of this first file. If I look at the end tail of this, I know that my last element is spectrum 28,740 from my third file, right? So now this P key, that is a concatenation of the file names and the spectrum identifier, uniquely identifies all the spectra in my raw data object. And so I'm going to do exactly the same thing for my identification data. So here's the code. And again, hat ID $primary key. Again, allows me to say, well, my first PSM, it comes from this file and this spectrum identifier. And so that's a way to be able to kind of uniquely make links between raw data and identification. Now, again, to simplify my task here, if I look at these, let me look at the first ones. Here they are. Look, actually, I have two PSMs for scan 15857 in my first file, and that's not really unexpected. The fact that a scan can match multiple PSMs. And just to keep things again simple here, because we don't have the time, I'm going to get rid of any scans that have multiple matches. There are ways around this, and it would be really interesting to look at these data into detail. But here, for the sake of time, I'm simply going to kind of cheat and simply take the those that match uniquely. And so what I'm now going to do is that I'm going to join spectra data, join information from my raw data, and join information from my filtered data. And both can be uniquely matched using the P key, primary key. So let me overwrite my object SP, and now check back into the spectra variables. And now I see that for each spectrum, I have my original information, but I also have information about the identification. And so here I literally joined the information from my raw data. And I've done it all manually. I mean, that's kind of the point here, to explore, to become very familiar with my data, to join these pieces of information. And so, yeah, there is an error, but I'll look at these later on. So why do you get 2 entries for scan number? Because I have a single scan that was produced by my mass spec, but I had 2 possible matches for this one. For example, if there is some uncertainty, there is not always a kind of unique matches. This happens. Actually, I didn't. It was for a minority, a small minority of cases, right? It happened that I had the case here at the very beginning and the second one here, but it was a minority of situations. Could you add the code later? I can add the code later, but I think the best thing to do is to look at here in this tutorial, right? And the links are in the notes. And here you have kind of documentation as to how to handle identification data, lots of examples. So all the information is available. And so now in this SP object, I have joined raw data and identification data, but I have done this in my SP variable, not in my experiment. So what I'm going to do here, spectra, is to overwrite, kind of the part of my experiment that contains the spectra. And so now in my experiment, I have the links to the files that are used and my raw data, but this time this raw data also contains the identification. So do you get 2 entries because the search allowed ranks 1 and 2? This could be a reason, but here the filtering that I did, where was it? Here, the filter PSMs actually only keeps only the first ranks. So you're absolutely right, Susanna, but in this case, it's for another reason. And so, so here we go. So I have this experiment, and I'll just I'll tell you to, if you're interested in this, look at the vignettes about experiments and raw data. There's of course so the MS experiment package and the Spectra package. There are many more things that can be done if you're interested in. But what I'm going to do now is that let me copy this over here. Right, I have, what is it, 87,000 spectra. Not all of these are equally interesting. So what I'm going to do here is that I'm going to say, well, now that I have I have joined my identification data and my raw data. Just do this, right? I have all the database accession numbers for the spectra in my raw data. So for example, I can see that this identification number contains 2 spectra. So this protein was identified by 2 spectra. This other protein was identified by 4 spectra. So what I'm going to do now here is that I'm going to create a subset of my raw data that only contains 13 spectra, with all the spectra that were matched to peptides belonging to that protein. Okay. So SP is my raw data object that contains 87,000 spectra. Now SP underscore 043175 only contains 17 spectra for that protein of interest. And very often, once you get to the very end of your quantitative analysis, your statistical analysis, you get some really interesting hits. It's often really good to go back at raw data and somehow make sure that your raw data, whether it's in terms of identification or quantitation, actually supports your final conclusions. And what I'm going to do here is that it's a little bit technical, but I'm going to kind of change one backend to another one. So everything I have done was actually really fast. Reading these 87,000 spectra into R was really fast. And that's because the data was always stored on disk. So I kind of saved a lot of time by not loading the data into memory, but keeping it on disk and only access it when it was really necessary. But now, given that I have a really small data set, I'm going to say load that full data set into memory, like change backend, so that now these 13 spectra, a tiny data set, are readily available in memory, and that can be very fast to manipulate. And why do I want to manipulate these very quickly? Because I will want to do some clustering. With the compare spectra function is going to take all pairwise comparison between these spectra. And I'm simply going to give them some names to make it kind of easier to look at. And it's going to, I can now easily create this heat map where each cell here corresponds to the similarity between these two these 13 MSMS spectra. Right, so the similarity is 1 along the diagonal, because here I compare one spectrum against itself. And then here I have one cluster of spectra that are very similar. They have distance between 1 and, what is it, 0.6, also 0.6, 0.7. But in here, I see that I have two subclusters. And they all have the sequence DLP something. So they have all been assigned to the same peptide. But I do see that within this peptide, I have two subclusters. And we'll have a quick look later on what this means. And here I have another cluster with peptide sequences that start with 2TQT. Okay. And here I have other two subsets of clusters that were assigned to the same sequence. But actually, I can see here that the distances are much, the similarities are much smaller, right? So here I have three peptides that have been identified or matched to the same peptide sequence, but actually they seem to be quite different. And we can have a quick look at this. And actually, maybe now is a good idea to have a look at this function here. F, yeah. So the spectra of this package has another function called browse spectra. And so if I call browse spectra, and I have a window somewhere, where is it? Sorry, where did it show up? Ah, it's here. It's here. So this allows me to kind of browse, navigate through my full experiment. So this is my first spectrum out of the 13. You see here, 1 out of 13. And that spectrum, here's the retention time, the precursor M over Z, was assigned this sequence. And here I have this MS2 spectrum with the matched fragment ions. And so here I navigate to the second one, the third one. And now that I have a kind of a small subset of peptides of interest, I can easily, that doesn't make sense for 80 or 90,000 scans. It would be possible, but it would not be very useful. But here now I can kind of navigate from one to another. And that's also something that is quite interesting once you have reduced your raw data to a subset of scans, spectra that you want to investigate in greater detail. And that's with the browse spectra function from the spectra of this package. But let me regenerate my heat map here. Where is it? Sorry, too many things open. Yes, of course, I first need to kill this. Here it is. So let's quickly have a look. We have a couple of minutes left. What happens to here inside this subcluster of peptides that were matched to the same sequence, but we see that we have kind of these two that are grouped together, and there's these three others that were grouped together. But they all have been matched to the same sequence. So what I'm going to do is that among I'm going to focus on those that have a sequence that starts with DLP. Right? These are those that I'm interested in. You know, the three first amino acids are DLP, and these are scans 2, 4, 9, 10, 13. And let's look at the information of these three using the same type of code that we had before, spectral data, now of my subset, and only the elements 2, 9, 2, 4, 9, 10, 13. And I'm going to look at the precursor charge. So all these precursors had a charge of 2, the precursor M over Z value. And here now I suddenly see that I have two different M over Z values for these precursors. And if I look at the modification name, I see that 3 out of these had modifications and 2 did not. So these 3 here, those that had modifications, and the 2 at the top here that form this other cluster are those that did not have any modification. And if I want to compare these in detail, I can also produce spectra, mirror visualization of these spectra, where I have one spectrum. So this one was the 4th one, and then the 9th one. Okay, to really kind of compare these spectra 2 by 2. Okay. So it was probably quite quick at the end, but what I wanted to demonstrate is that even though we can load this large data set, and here I only use 3 files, and this will easily scale to tens or even hundreds of files, again, because we use clever tricks to avoid overloading the memory of our data set. You know, what would take the most time would be to download these 100 files and not to load these 100 files in R. But sometimes we can focus on some scans of interest. Here I chose the 13 scans from a protein, and then I can cluster these MS scans based on distances calculated using the fragment M over Z values. And we kind of focus on one subset of these and identify that we had 2 subclusters with and without modifications. But these are the kind of things that you could do, or kind of selecting any scans of interest and not necessarily only those that are identified. So can you also load top-down data and use these packages? As long as you can export your data to ML files, then yes, you will also be able to manipulate these data. You will also need to have identification data. And again, if you can export these to NZID files, then you should be able to do everything I described with any type of data. The only requirement is that the data need to be exported in one of these open formats to be able to handle these. And so also a really good question here. If the purpose of this tool is to record how MS data is analyzed so that it is reproducible, at what stage would we implement it? Right after searching and identifying proteins or before additional analysis? Also, how can we incorporate other tools like MSStats, TMT into the script? And so this really depends, what are you aiming for? So here I focused on raw data, but you could proceed with that analysis, with that concept of an experiment, and also add quantitative data that you would use for your statistical analysis, for example, using MSStats, TMT, if you have TMT data. So really that very much depends on, where do you want to invest your time to make sure that you understand and are in a position to reproduce all the steps of your analysis. Sometimes, like I showed here, this could be at the very beginning when you have the raw data. But in other cases, you might not have access to the raw data, or you might want to focus on the quantitative data and on the statistical analysis, for example, using MSStats TMT. And so in that case, you could start later on. I think that will very much depend on what your priorities are. There are cases where I start from the beginning, as I showed here, and there are cases where, for example, I get a spreadsheet with quantitative data that was produced by, I don't know, MaxQuant, Protein Discover, any piece of software. And then I would kind of focus on the reproducible aspects later on. This really depends on your priorities, what is important for you, and also, of course, on the time you have. Can you invest time to go back to the raw data, or do you prefer to invest the time and your skills on the analysis of the quantitative data? Voila. So I see it's 6:30, so I think I have used all the time that I was allowed to use. And so I will add all the code that I wrote here to the tool page so that you have access to it. But kind of none of this code is new. All that code is available. Where is it available? Let me check. Is available here for mass spectrometry docs. Yeah. Is available in this tutorial with lots of details, such as introduction about kind of how does mass spectrometry work. But I'm sure all of you know this. Accessing data. How can we access data? And here's an example of using the RPX package. There is a chapter on raw data, right? Raw mass spectrometry data, and you will recognize the figure that I used before. Description of the spectral class. Much more about visualization, right? How to reproduce this figure, where we kind of delve into one MS1 spectrum in detail, and then look at all the MS2 spectra that were produced by selecting 10 precursor ions here. There is a section about identification data, quantitation data, and so on. And these tutorials are being updated on a regular basis. Okay, so I will look at errors that were posted in the chat and in the Q&A, and kind of try to address this directly in the chat. Otherwise, I hope this was useful, and thank you very much for your interest.
Thank you very much, Laurent, for the presentation. Demoing things live like that is never easy, and Laurent is really an expert in that. Thank you so much. So we now have time for a 10-minute break. Please take this time to take a break, but also to ask Laurent additional questions in the Q&A. So Laurent kindly agreed to stay for a little bit of time. Well, it's getting late in Europe, but Laurent will stay a little bit